by the National Science Foundation through the ATE program. We encourage and support ideas and innovations you have for educating biotechnicians and encourage you to consider applying for ATE funding. Please feel free to reach out to us. Before we begin, I have a few housekeeping announcements. This presentation is being recorded and the video will be posted on the innovatebio.org website by next Monday afternoon. Both of our presenters have noted that during the presentation, there will be a brief pause for a short Q&A as well as the end of the presentation. And lastly, additional information about future ATE project talks can be found on the Innovate Bio website, which we will put in the chat. Thank you. So it is with great pleasure that I introduce our ATE talk speakers. Dr. Margaret Bryans from Montgomery County Community College in Pennsylvania, and Dr. Lori Kelman from Montgomery College in Town, Maryland. Their presentation is titled NISTCHO, Cells for Teaching Biomanufacturing and for Undergraduate Research. Welcome, Maggie and Lori.
Thank you, Dion. Can everyone see the slides?
Yes, but it's your preview screen.
Okay, got it. Just switch that over. Oh, just try this. I'm going to stop sharing and pick the other screen. Just 1 second. Better?
Perfect.
All
Thank
right.
you so much for the invitation, everyone. Maggie and I are going to tell you about NIST Cho cells, which we hope you're going to adopt into your biomanufacturing programs, your biotech programs, and if you're interested in performing undergraduate research. And this work was supported by NIMBLE and funds from the American Rescue Plan. So this slide is going to show you what we're going to cover today. There's a lab-based curriculum that's been developed to get NISTCHO cells into your biomanufacturing and biotechnology courses and your workforce development programs. As you'll hear, we've got SOPs, batch records, and some representative data that we're going to show to you using NISTCHO cells and which produce cNIST mAb. And we'll tell you about some of the outcomes of our undergraduate research projects. So I'll start with a brief introduction to NIST, the National Institute for Standards and Technology. This is a part of the U.S. Department of Commerce. It's a non-regulatory agency. I like to call NIST the people who bring you the correct time and the standard kilogram. But they have standards and reference materials for many industries, most actually not biologics, which is somewhere that they're fairly new to. These reference materials allow for industry-wide calibration and comparison, and shown here, one of my favorites, the NIST standard cigarette, which is not for smoking, but for ignition testing. And I'm not quite sure who's using the NIST well blubber reference material, but there it is. Reddit had a field day with that. And the standard peanut butter. But we're going to tell you about NIST Cho cells and a little bit about NIST mAb and cNIST mAb. So the NIST Cho cell line, this is a fairly newly released recombinant cell line. It became available a year ago in January, and it produces a non-originator version of an anti-RSV monoclonal antibody that we're going to call cNIST mAb, and the little c stands for Cho. The NIST Cho cell line was developed by NIST and produced by MilliporeSigma, and it's currently not available as a reference material, which is coming soon, but currently it's available as something called a research grade test material, and I'll say more about that in just a moment. NIST is a very well-studied monoclonal antibody reference material that was produced against the RSV protein F. The epitope is shown here, and NIST Cho cells produce, as I said, what we're going to call cNIST-MAB. And there's a lot written about NIST-MAB, but we're expecting a publication soon on NIST Cho that will include the details of strain construction. Next slide, please. But you can get your NIST Cho RGTM cells right now. So the research grade reference material designation is for the community to work with the material and collaborate with NIST, letting them know if the material is suitable for purpose. And the idea here is typically they're going to ask people to perform research and do other things with the cells. And then once a year, one of the requirements of using the RGTM is an annual report to NIST. This year it was due on March 1st. And NIST has told me verbally to encourage colleges to use NISTCHO in teaching. So we have a slightly different I don't think they're expecting us to do lots and lots of research, but fit for purpose is still going to mean, did the cells arrive okay? Did they recover? Did they perform? And if we're using them in teaching, NIST would like to know about that. So I'm going to encourage you to get your NISTCHO cells now. Next slide, please. And Sandy, if you would put the first link into the chat. So this link will take you to a little bit of information about the NISTCHO RGTM. From there is a link to the NIST store. You would have to create a login. But one of the benefits of the RGTM is the cost is zero at this moment in time. NIST will even pay for the shipping to send the cells to you, and they will arrive on dry ice. But as you'll hear in just a few minutes, we have evidence that the cells could be stored at minus 80 degrees for several months. So if you don't have liquid nitrogen storage and you're not able to immediately thaw the cells, I'm going to tell you tentatively, I think that's going to be fine. But what we're going to encourage you to do is get the cells, expand them, make your own bank, and then store them so that you can teach with them. And the cost of the reference material is not out yet because it hasn't been released yet. But if it's like the NIST-MAB, the cost will probably be about $1,000 per vial. That is a recovery cost through NIST, but the NIST Cho was made essentially under GMP conditions. So it's going to probably cost more than zero. Next slide, please. So once you receive your NIST Cho cells, and this is where you put the second link in, please, Sandy, there is a manufacturer recommended protocol to thaw and to grow the cells. Now, the link that Sandy's about to put in is a 33-page manual for chosen cells. This is the host cell that was turned into NIST Cho. But we're going to release our own version of protocols. And one of the reasons is because you don't need all the information in the longer handbook. And in particular, you do not need glutamine in your media. So the recommended media is going to be XL Cho Fusion, and that's for maintenance or what we're going to call expansion of the cells, and XL Cho fed batch media plus supplement and glucose for production, and we'll call that production media most of the time. Now, we have some evidence, and we've heard from others, that NIST Cho cells will grow in any other media that you could use to grow Cho cells. So if you don't use this media, again, not a promise, but I have a feeling it will work just fine. We're going to share, not today, but online, we're going to share thaw and recovery SOPs, sample growth data, instructions for making your own bank, and also, and you'll hear about this today, biomanufacturing SOPs and batch records. So our suggestion is that you should get your NISTCHO RGTM while they're still available. thaw and recover them, expand them, bank them for your own use, and then start to use them when you're teaching biomanufacturing or bioproduction. Now I'll turn it over to Maggie.
Thanks, Laurie. So MC3, we adopted the NISTCHO cell line this semester actually for teaching a biomanufacturing course. We had been using the cell line since August, developing the directive documents with our student interns, SOPs and batch records, etc. So we're replacing a cell line we had been using since 2016, and that was the Cho DP12 cell line. And we chose that to develop the curriculum with because it was available from the ATCC. So anybody using the MBC2 curriculum could also have access to the cells. However, ATCC stopped providing this cell line, so it was really serendipitous that NIST released the NISTCHO cell line early last year, and then Laurie and I came together to conduct this project. NISTCHO cells, as Laurie said, are derived from the CHOSEN platform. They grow at much higher densities and demonstrate much higher titers than the Cho DP12 cells. We're actually very, very happy with the cell line, and I encourage you to switch over if you have that capability. They're great to work with. We've been developing upstream and downstream processing, the directive documents for the curriculum over the last several months. We've grown the cells in tea flasks and various sizes of shake flasks for production, and also a 1 liter culture in a bioreactor. So that's what I'm going to talk about next, basically what we've been doing in the biomanufacturing course and how you can adopt NISTCHO in your biomanufacturing and biotechnology. You can even use it in your intro to biotechnology course as well. At this point, we don't have a functional assay such as an ELISA, but we are working with other groups to develop that. And we're also working to have a better assay for titer. And we're doing sort of an analytical protein achromatography on the HPLC to get better titer measurements as our approach. There's other approaches to do that as well, but they require more sophisticated instrumentation. And we already work with our HPLC, so that's the direction we'll take. So when you start to grow the cells, so this is a 30 ml culture in a 100 ml flask you're seeing here. And this experiment done by one of the interns was a comparison of how the cells behave in the two types of media. So that XL Cho fusion media that we call expansion medium, and then the production medium with the added feed. So the feed actually you purchase separately, and they recommend you add it between 5 and 10%. And the reason we changed the name of the media to use it was because if we're doing a straight batch strategy as our upstream processing, we don't want really the word fed batch in there when we're not adding additional feed. So we call it production media. We developed 2 SOPs here, as you can see, for initial resuscitation and culture of the NISTCHO cells. Those will be available to you, and also culturing in shakes, flasks, and subculturing for monoclonal antibody production. And on the right, you see a couple of growth curves. Again, in blue there is the cells grown in the expansion medium, which typically give us a maximum cell density of about 6 million cells per mil. And with the production medium and the added feed in day 0, we see a much higher cell density of about 14 million cells, and we also see higher titers as well. So you can see the viability is great throughout until the run out of glucose, and then you see that decline. So very reliable cells. This is very reproducible, very easy to grow with. And then we scaled up to 100 ml culture. So this is an experiment done in duplicate with 100 mils culture, in this case in production medium without the extra feed. Cells were seeded, or the culture was seeded at 3 × 10⁵ cells per mil. Maximum density reached was 1.4 × 10⁷, and our titer estimate is about 0.5 gram per liter or 0.55 mg per mil. Right now, we're determining titer by actually purifying the mAb from the clarified media after harvest, and then measuring the concentration of the pure monoclonal antibody after protein achromatography using a nanodrop with the IgG function to get a concentration.
So Maggie, there's a
Yes,
question in the chat from Leah.
yes.
How often did you add the feed, and was your 5% by volume?
5% is by volume. In this case, the experiment I showed you in the last slide, we added it only once right at the beginning. So we're actually working, and we've done several sort of a fed batch strategies where we're adding it in day 3, 5, 7, 9, and so on, and we can take it all the way up to day 14. We're still working out the actual and getting some reproducibility with that, but for right now, in that experiment, the feed was just added at the beginning, and it's 5% volume per volume. So you could do your whole experiment small scale, and what we like to say is low tech. You know, you can introduce this in your biomanufacturing course without going to a bioreactor. You could scale up in shake flask. So this is a strategy here where you can thaw your NISTCHO cells, resuscitate, grow them in a 30 ml shake flask, maybe scale up to 100, or we have data for 200 mils, and then harvest in a benchtop centrifuge, followed by 0.2 micron filtration. And then you have your clarified media, which you can then purify the MAB using capture chromatography with protein A resin in a gravity approach, and then have your students generate the chromatogram. So that's something we did with the Cho DP12 cells as well, and then they can assess the purity with SDS page. So this is very accessible to most people. That they have CO2 incubators. You know, they don't have to have sophisticated instrumentation to take this approach and adopt the cells to make monoclonal antibody. Here we see one of those gravity columns, 1 mil resin with, in this case, 5 mils of media, clarified media. And here's the SOP of this sort of a low-tech purification of CNS map using gravity protein A. On the right, you see a representative SDS page run under reducing conditions, and the expected MAB fragments, the heavy chain and the light chain, can be seen here. So this is the pre-column material, and since it's a chemically defined serum-free media, there's not a whole lot of other proteins really there. That you can see the heavy and light chain are well represented, even in the unpure media. And then after purification, you can see elution fractions 3 and 4 here with the purified heavy light chain. And again, we have a fairly pure preparation here already, maybe in the 90% purity. So again, this is the low-tech approach, very easy to adopt and use in your course. In our biomanufacturing course, we do go from the 100 ml culture all the way through to the bioreactor. So what you see here is kind of a flow chart of our upstream processing and downstream processing. And again, this is following our SOP and our batch records, which I'll show you in just a second. So again, the 100 ml culture is inoculated with 2 vials of frozen cells from our working cell bank. And the students then monitor the culture for about 6 or 7 days. Monitor, I mean, they take a sample aseptically from the culture, and they measure optical density at 650 nanometers, pH, cell density, cell viability, and glucose and lactate levels. So they get experience measuring those parameters. They grow the cells to approximately late log phase, which is between 5 and 6 million cells per mil, and then scale up to the bioreactor with a seeding density of 4 times 10 to the 5 cells per mil in production media. So now we really want the cells to produce, so we move to that richer medium. In this case, it's a straight batch approach, so we're not feeding along the way. But we do increase the titer by switching to the production medium. The production medium costs about twice as much as the expansion medium, but it's nice to start in the expansion medium because it really gives them sort of that real world feel of you're expanding the cells in the seed reactors before you get to your production reactor with all the goodies, so the cells will really perform for you. So the bioreactor is sampled, and pH and DO values are recorded in the batch record because these are process controlled with probes in the bioreactor. And then all the other parameters mentioned above are also tested for and captured, results captured. After the bioreactor run, we take that to usually about 5 million cells per mil we get as a maximum density in the bioreactor, and we can talk about why that's lower than the shake flask in a second. We take it out to day 8 or 9, where the cells have reached plateau or early decline phase, and then harvest by centrifugation, basically taking the culture, putting it into centrifuge bottles and collecting the media, which we then clarify by 0.2 micron filtration. The next step is very optional because the titers are sufficient with this cell line. You can go directly from the bioreactor clarified media right onto the protein A column, and you'll still get a really decent peak, as you'll see in a moment, of MAB when you purify it. I, at this point, introduce tangential flow filtration just to give the students experience with TFF, and we have a benchtop unit, and it helps them understand that's an important part of UF, ultrafiltration, diafiltration in the biomanufacturing process. And then we're on to protein A chromatography on the ActaPure system in our case, and then with optional additional polishing chromatography with ion exchange, and then assessing the MAB purity using SCS Page. So that's basically the upstream and downstream we just went through with our students this semester. And what you're going to see next is the data that they produced in the course. So again, this is representative data. We've run these experiments now, especially the 100 mil culture experiments, many times. So this is definitely reproducible. Again, what you're seeing here and the parameters I mentioned earlier that we test for, we see our growth curve here in blue and taking the cells to late log because we want them to be in good shape for seeding the bioreactor, and then very consistent viability over the 7 days. And then here we're looking at glucose usage through the growth curve and the decline in glucose here, which is to be expected. Optical density, which measures turbidity of the culture, as you can see, mimics almost perfectly the growth curve. And here you're seeing pH measurements. So this is by taking a sample and measuring with a pH meter and a microprobe to get our pH. You see a dip here, but it stays within sort of the physiological range. Here are the SOPs there's an SOP for the entire upstream process, which includes the 100 mil and also the 1 liter bioreactor culture, and then the full batch record that the students are completing as they go. So this is a paper batch record at this point, and they're completing it as they go and introducing all the test data day by day. So it's about a 3-week process for the upstream processing. And then they move on to the bioreactor. These are 2 students from the course here. We have an Applicon traditional bioreactor with a glass vessel, and it's a 3-liter vessel. We do a 1-liter culture in there. And again, this is in production medium and a straight batch strategy where we're inoculating and letting the cells grow through the growth curve and then harvesting. I mentioned already the seeding density, and we went through several iterations of these bioreactor runs in the fall, just really trying to get something that was very reproducible. And these are the parameters that we are working with now. And we've done 3 runs with these parameters, and it has been very reproducible. We're at 40% dissolved oxygen, 150 RPM agitation, pH 7.1, and the addition of antifoam as needed. In this case, 0.5 ml in a total of 10 mils of media are added usually about day 3 and day 4, and perhaps again on day 7. So that was something we had to really spend a lot of time working that antifoam issue out. And again, here are the 2 directive documents.
Maggie, can you repeat the brand for the bioreactor?
Yes, Applicon.
Thanks.
So here are the data for the bioreactor run and what you can expect under those conditions. And we see here our growth curve. going through exponential and then plateauing here. And here we can see the effects of actual foaming, where we added our second dose of antifoam here, and then we got the cells to, you know, uptake a little bit. Very good viability. And then again, optical density as we go through. We took it to, as the cells approach decline phase in day 9 before we harvested. We've also harvested in day 10, and we actually get a higher titer if we harvest on day 10. Here, because it was part of the classwork, we harvested in day 9 because that worked out best for the schedule. And then we see glucose usage here as we go through the growth curve, and as well, and here you see our dissolved oxygen process controlled around about 40%. And part of the reason we do get foaming is because these cells grow very well. They grow to much higher densities than we're used to, and we operate on clean air rather than pure oxygen. So the system has to really pump in quite a bit of air to meet the oxygen demands of the cells. So then we have to deal with the foaming issue as that arises. But of course, antifoam is commonly added to bioreactors in commercial scale manufacturing too. So it's very real world. So those are the results and consistent results we've had with these with this approach and these parameters. And this was generated by the students this semester. After the bioreactor run, we then harvest, as I mentioned, and then this is a student using our benchtop TFF unit for an ultrafiltration cassette there. It has a 30 kilodalton cutoff, the filter within, and then the SOP that goes along with the harvest and end of run. Then on, as I mentioned, to protein A chromatography and our SOP for purifying CNS mAb, and here you see a representative chromatogram. So in this case, we took 5 mils of medium directly from the bioreactor. So this gives us some idea of the titer, not concentrated, and applied it to the column. And you see here our flow-through peak, and then with the addition of our buffer B here, which is a low pH citrate buffer, we see our elution of our mAb. So the protein is just a wonderful resin. It works great every time. And we see our mAbs pretty much there in just a 1 mil fraction. Just to wrap up, this part up is these are the SOPs that we've developed for using for the initial culture and resuscitation of NISTCHO cells, then shake flask culture and a 30 mil culture and subculture. And also the growth of the cells, and you'll see some evidence of that in a moment, in the T75, where you can, even in one T75, get detectable monoclonal antibody with gravity chromatography. Then the entire SOP for the upstream process is the batch culture SOP, and then the batch record, and then downstream processing, end of run, purification with gravity flow, and also with ACTA, the ACTA system, and the batch record. So we'll be in the next week or so adding these SOPs to the MBC2 site at biomanufacturing.org. The link I have there right now is the curriculum we have for the Cho DP12s. Obviously, we will be adding and recommending the switch over to the NIST Cho cell line, and all of these SOPs and directive documents, as well as material lists as well, will be added to the site. So in addition to creating directive documents for others to use NIST Cho cells in their biomanufacturing training, we also, as part of this project, wanted to use NIST Cho cells in undergraduate research projects. So both Laurie and I had students last semester and this semester work on many aspects of the cells, and I'm going to hand it back over to Laurie now to tell us what happened at Montgomery College.
But before I do that, if anyone has a question at this moment, please feel free to unmute and ask or put it in the chat. I'll share that we did not use NIST Cho in our biomanufacturing course this semester. We had hoped to, and we have ongoing single-use closed system 5-liter bioreactor work, but we have been unsuccessful in getting a good run. So, but we have been successful with some undergraduate research. So I promise you, you are going to love NIST Cho cells. We hope that you're going to adopt them for your teaching, but you're also going to be able to adopt them if you're performing undergraduate research. Maggie, if you would change the slide. So you'll find that we took very different approaches. At Montgomery College in Maryland, we have a scientific research course, and over 3 sessions, I had 22 students, some of who took the course multiple times. And what they were working on was growing the cells, looking at different seeding densities. I'm going to show you some cryopreservation data. And a lot of what they did also was looking at the MC3 produced SOPs and beta testing them. And we found it in our hands very easy to adapt these SOPs to our equipment. So again, I'm going to tell you that I think you will be able to do the same. And if you go to the, yeah, go ahead.
Sorry, I want to interrupt with a couple questions before you go on, Lori.
Yeah, sorry.
It's all right. One is from Linnea, and then Leah, you can ask yours. So Linnea is wondering if industry is interested in the Cho cell line.
So the reason that the NIST Cho was produced was for industry and by request of industry. After the NIST MAB was released, which was either 2016 or 2017, I'm pretty sure, industry said, oh, this is great, we love it. Could you please give us the cell line that made NIST Cho? And the answer is no. That was a proprietary cell line by what was at that time MedImmune. And they said, nope, you're never going to see that cell line. Would you please make us a cell line then? And NIST responded, they made an NS0 cell line, which was the original producer cell, but industry said, we'd rather have Cho. And then the development of NIST Cho started. And I saw Brad in the audience. If there's anything you'd like to add, Brad, please feel free. And so again, first they did the release as the research grade test material. When they released the RM, the reference material, now I'm not sure about this, but I believe the RGTM will go away, which is part of why we're encouraging people to get it now while it doesn't cost anything. Did that address the question?
Yeah. Before you ask yours, Leah, I'm going to give you one from Crystal Auger, who just wants to confirm that the cell lines are available for career and technical high school biotech programs.
So what I was told by NIST is that anyone can request the cells. There are some conditions to the cells, and for example, they're not going to ship to your house. But I don't honestly know about a high school, but feel free to like put a little information into your request. And I'm pretty sure the email is nistcho@nist.gov, which I'll stick in the chat in a second.
Oops. Okay, and then Leah.
Yeah, I was wondering about the extinction coefficient that you used for your nanodrop measurements.
It was for IgG, so we used
Just like a generic, like.
Yeah, just a generic, exactly. It's just, you know, from at this point, we say it's an estimate for the titer because we
15
can't.
or something.
Yeah, exactly. So just what's kind of programmed in to that.
I think the default is 15. I don't remember. Something like that. Okay. It's
published for the NIST MAB, and I was just trying to find it very quickly and I can't, but NIST MAB is certainly not going to be precisely the same thing as the NIST MAB, but once we get a little more under our belt, we'll probably switch to using that instead, although we haven't investigated that at all.
Okay. Also, has anyone in this group yet tried using an LC method for titer? I actually just talked to Kat this morning because I think we're going to try to set up a CRADA with NIST to like do a deeper dive on some of this work just to make the information more widely available, both to researchers and also to industry. But
I think Brad, who's here, I think has been working on maybe even the porous method. Brad,
Okay.
I know you were embarking on that at one point for titer. No,
So
he's not
we'll
available.
start working on it probably in the next month or so. But Brad, hi Brad. Okay, that's fine. I was just curious because we're
Maybe Leah,
not
can you
interested
share?
in starting totally from scratch. But we did
Mhm.
have a shake flask run where we did feeds with all the same media as Maggie is what you did, except that we fed every other day about 2%. And we
Mhm.
same cell densities, but we haven't measured titer yet. I'm still waiting because we don't
Mhm.
have a method up and going.
Yeah,
And I
So
didn't feel
it's
like
nice to
buying
get.
and packing robocolumns. So
And this is perfect for teaching, right? What we have, we don't have the sophisticated LC instrumentation, but this is great for teaching, for them purifying the mab and doing the
right.
concentration, and then understanding that it is an estimate, but it gives us something to hold on to. It gives us something to compare.
Leah,
Okay.
can you share your interest in the NIST Cho cells?
Can I share mine?
Yeah,
Oh yeah.
what do you
So
do with them?
well, right now I'm just growing them. So yeah, I'm working for Waters, and Waters is interested in process analytical technology. Of course, they want to build IP, but they want it specifically for upstream cell culture. So right now what we're doing at the Merce lab is working with the NIST Cho and the CNIST MAM to get a process up and going. The idea is that, we would do process development, including some basic process characterization, possibly for multiple processes and various platform medias that are available. But what Waters would want to do then is, like spent media analysis and AAA and peptide mapping and stuff on their instruments, partially to support collaborations with other industry partners, but also just for their own education. And this is what we have. This is really the only cell line we have to work with right now. So we're just going to focus on it because me personally, I think there's a huge value add. It's a huge value add to the community to just focus on this one. Waters has no interest in developing proprietary cell lines and molecules, so that's not our business. So if we just work on this, we could get a lot of really useful information, including the potential of developing, a chemically defined open source media recipe for this cell line, right? Which would be awesome. You wouldn't have to pay for all these commercial medias. So that's what we're doing.
Thanks. We have one more question before. Sorry for the interruptions. Donna Barron is asking about the HPLC method. I believe and Donna, is this the one that you're talking about that Maggie's been using?
No, yeah, for
Oh.
the titer determination that you guys were just talking,
Mhm.
maybe I missed something. We have HPLC here, we have UPLC here at
Mhm.
HVCC, and we'd like to incorporate that into our biomanufacturing course, and not just determining aggregate content, but for that titer method to
Mhm.
do, in process sort of testing. So if anybody has a titer method for using the HPLC, I'd like to, if they would be willing to share that, I would be really interested in it.
So Donna, we're developing that method, and that's something we talked about at the hackathon as well. And we had really good industry SME input. So I have a really nice sort of a white paper I can share with you
Oh, that'd be great.
on how you do that. And as I mentioned, we're still in the early stages of doing that as a more accurate method for titer determination. But I will share that paper with you. That's the best I can do at this time.
Thanks, Maggie.
You're welcome.
Okay, back to you, Lori. Thanks.
Next slide, please. So these are the students who performed undergraduate research at Montgomery College. And as you can see, there's a bunch of them. Ryan has an asterisk next to him because he converted to a part-time student worker. And in the next slide, we had some faculty and staff assisting as well. And Ananth was hired as a part-time laboratory specialist. He performed some of his own work, and he also helped out a great deal with the students. Next slide. So one thing the students got was tons of experience counting cells. We performed manual and automated cell counting, and I'm not going to show data on looking at seeding densities. So almost everything we did when we grew the cells, we did growth curve and checked for viability and density. But I can summarize, well, a couple of things. One is, first of all, it's undergraduate, so time points were not always strictly 24 hours. And in particular, we weren't really trying to optimize. So we wanted the cells to grow, but although we tried an awful lot of things, we did not do it as systematically as one might hope. What we did find in general, though, with seeding concentration is thinking in terms of somebody who's teaching. You can seed NISTCHOs to get yourself a culture in 3 days or 4 or 5 or 6 or 7, no problem. The cells are really flexible. They seem to understand that we have teaching schedules and weekends and holidays, and so I think you're going to be very pleased with that. Next slide, please. We tried growing the cells a bunch of different ways. So we grew them in the expansion media. We grew them in the fed batch or production media. We added feed. We added twice as much feed, 5 or 10%. We added 5% or 10% glucose. We tried a number of different things. And to summarize, NISTCHO cells grow really well. If you supplement them, they will grow. And if you don't supplement them, I don't think we tried fed batch without supplement, but I mean, you can grow them kind of any way you want. Shown is a growth curve. And one thing I will point out is you'll notice both growth and viability, you get this sort of precipitous drop, which we just keep calling the crash. And we noticed it, Maggie noticed it, other people have told us it happens. So just be aware that when these cells reach whatever moment they've decided this has to happen, they will just crash on you. Next slide, please. One of the SOPs that we beta tested was a T75 flask growth and CNIST mAb production. And so realize that these are cells that grow fine in shake flasks, but here we just put them into a T75 and we tried this in both production and expansion media. And sure enough, they grow. So if you want to go truly low tech, if you don't even have a shaker, turns out they'll grow this way. The growth was followed by microscopy. As you can see, from day 0 to day 7, the cells grew. And we found that the SOPs were very repeatable and you could grow them. And on the next slide, they produce CNIST mAb, or at least something that looks like it should be it. The students, we followed production by absorbance at 280 and many Bradford assays. And the gel shown here is not specifically from the T75 experiment. Sorry about that. But the blue arrows show the eluted protein that's got the right size, unreduced and reduced, as would be expected. And another thing I'll mention is we took our conditioned media. We did not add protease inhibitors. And sometimes we stored that media more than a week at 4 degrees before we purified the protein. And again, these cells seem to understand that we might be teaching and might not have lab every day. Next slide, please. We could purify CNIST mAb from all sorts of NIST Cho cultures. The purification was very straightforward using protein A chromatography, whether it was a gravity column, whether we took those columns and spun them in a centrifuge, or whether we used our Acta Avants. And again, it was easy to take the MC3 SOPs and adapt for our equipment. Next slide, please. So another thing we did was take a look at how the cells could be cryopreserved, because we thought that people would like to make their own cell banks and hang on to them so that they didn't have to order cells every semester. So we made the cell banks standard fashion. I believe we banked in 7% DMSO. But then what we did was cells were stored at 3 different storage temperatures. We have a mechanical -150, which is seems to be equivalent to liquid nitrogen vapor phase storage. And that, of course, is how you're supposed to store them. So we stored them there. But we also stored cells at -80 with the thought that not every college would have liquid nitrogen storage. So we stored them there. And even though every textbook you've ever read said you should not try to bank cells at -20, we stored them at -20. And showing just a tiny bit of data and obviously not particularly quantitative. But what we found is that you can, so the NISTCHO cells can be stored at -150 and thawed and recovered as expected. You could store them at -80 and it looks the same. You cannot store them at -20, which was not a shock, but again, we did want to see that. And if you look at the next slide, the way that we thawed when we did this was the cells were thawed and grown in culture for 7 days. And if they were still looking good at that point, they were passaged once, just so that we could show that they really truly were okay. And this is just a single, just one of the bits of data. But in blue, the -150 stored cells have a growth curve. Same thing in orange if it's -80. The -20s stored cells never really looked good. And then they, forgive me, crapped out after about 2 months. But we've got data through week 21 post-banking, and you can store them for several months, and Maggie will have more to say about this. Another thing that's happening here is not all of the students performed wet lab work, and so I have students who have determined doubling times from all the growth curves from all the weeks. And the preliminary is that we don't see a difference whether you store at -150 or -180, the cells seem equivalent. So again, we're going to tentatively recommend that you get your cells, bank your own cells, store them however you can store them. If they're at -180, you may have to make another bank. And with that, I will turn it back over to Maggie.
Thanks, Laurie. So at MC3, our approach was to use our biotechnology internship course. So over the 2 semesters, fall and spring, we had 4 student interns, and Shanna and Kayla were the fall semester, and they initiated the cryopreservation study, did some of the early bioreactor runs, and the stability study and the media comparison was done by Kayla. And then this semester, we have Todd and Seaham who are continuing those studies and helping us with the larger scale flask, shake flask cultures and finessing the MAB purification part. At this point, I'd like to also acknowledge Heto Doshi, who works with me at MC3 to develop curriculum, especially on this grant and also other ATE grants as well. So just real quick, because Laurie's already presented some cryopreservation data, these charts show nicely the comparison between growth curves, and this is in expansion medium, of cells growing, of cells cryopreserved up to 26 weeks. So we took the cells at 2-week intervals and put them into a flask and monitored their growth. And as you look at both of the charts, you can see they're very comparable. The yellow dots are the 26 weeks, and the sort of a darker blue dots are the 2-week stored cells. Excuse me. So you can see they're very comparable. That they actually freeze very well at -80. And not only do the growth curves look good and the maximum cell densities obtained with these cells after resuscitation, we also see very comparable titers. So these titers are low because, again, this is an expansion medium, but they're very comparable between cells in the 2 storage conditions. One of the other long-term projects we did with the interns was a stability study, and we were able to come up with this project as part of the hackathon with Sandy's antibody engineering grant. And the way we designed it was the student would resuscitate the cells and keep them growing in culture over many doublings, even up to 100 or more doublings, and passage them every three days or so. And then every 2 passages, she would freeze them down, or they would freeze them down. So we would have frozen cells at increase in passage number. We started at passage 12, I believe, and then we froze cells at passage 14, 16, and so on, all the way up to passage 32 at this time. So after freezing the cells, we then resuscitated them individually and grew them, and then had a look to see what their growth profile was, and also the titer by purifying again a fraction of the media and measuring the A280 of the IgG. And here are some of the results. So you see here on the top right is the growth curves of 4 of this is 4 examples of the growth curve, but really they are all very, very similar. Just didn't want to crowd this graph. So passage 10 was the initial passage number that we started with, and we see that kind of in the dark blue. And then the green is passage 32. Sometimes we don't have a data point because it falls on the weekend and the interns didn't come in the weekend. So, but you can see the overall pattern of the growth curve through 8 days is very similar with these cells, whether they be passage 10 or passage 32, and similar of viability. And then, you know, as they go into decline rather sharply with the depletion of the glucose. And then here I just wanted to point out the peak cell densities that we got for the cells, whether they be passage 10 or 32, and also again measuring the titer. So this is in production media without any extra feed, but you can see this uniformity of the titer with the new cells versus the old cells. So really we've gone over about 100 doublings at this point, and we still see the cells as being stable as far as their growth profile, as well as the titer, the antibody that they're producing. So that this is an ongoing study. Moving on, I would like to have some acknowledgements, and really I'd like to say Laurie and I would like to say thanks very much to Brad Odell, who was our subject matter expert from NIST, and he joined our project meetings. He was incredibly helpful as we started getting the bioreactor runs going, and just gave us really great expert advice along the way. Also to John Shield, Technical Program Manager at NIST. Namata Raman is our Program Officer at Nimble, and we met with her monthly. And again, just to acknowledge the funding from the Department of Commerce through NIST in collaboration with Nimble, and also to acknowledge the NSF ATE grant developing classroom-based undergraduate research experiences in antibody engineering that really got us going in these research projects. So thank you for your attention. And I can stop sharing if there's any questions or.
Yeah, and there will be a NIST Cho team at the hackathon this summer. There will be a hackathon August 5th through 8th. So if
the hackathon open for other participants, I have a tech starting that I think it would be great to assign her as one of your team members. So you could have her probably like 40 or 50%.
Totally. Yes. Yes, I will be sending. So we have a mailing list that is at the Innovate Bio website, and it's also at antibodyengineers.org. And we have not opened up the applications yet, but we're planning to open them up April 1st. But if you get on the mailing list, you'll get an email when it opens up. Yeah, what is a hackathon? A hackathon is not just coding. A hackathon is a group of people who are involved in a short-term, intense experience to create a product of some kind. And I will be talking about hackathons April 5th. So you'll be hearing more about them. They can be virtual, they can be in person. Ours are mostly virtual, but there are in person. Sometimes we have people doing in-person work as well. Are there more questions?
Well, I would like to thank you for presenting. I appreciate that.
Yeah, thank you. This is super helpful. And I don't have any data to show today, but we did just generate a shake flask study that pretty much corroborates everything that was shown today. Sans the titer, which we're still.
We've tried growing them in F12 with, I think, 10% serum and EMEM with serum and other things, and they grow. You know, and they were made animal free, so adding serum is not necessary. But if that's how you grow your cells, it should work.
Yeah, we also did something interesting where we froze the cells in large volumes. We put 250 mils into a 500 ml bag with the 10% DMSO. And they thawed amazingly well. We didn't do a follow-up growth study, but like the viability coming out of thaw, that was after storing at -80 for a week. But part of the reason we were doing that is because, you know, we're trying to do studies across multiple sites with cells, and getting them back and forth is a little painful. So we wanted to be able to ship them on dry ice.
So do you store it all at -80 other than what you just said? Because that was news to me.
I have
Maggie's
not
idea.
NIST Cho, but I have experience storing other Cho cell lines, like the Horizon Cho K1 cell lines at -80 for up to a year.
I was going to say I had an inadvertent experiment that NIST shipped on a Thursday, and the shipper couldn't deliver, and so everything thawed. So, and my thawed cells still had 30% viability, so these are pretty tough.
Wow.
Pretty robust, yeah.
Yeah, when I was having the first discussions before this project really happened with some of the NIST folks, you had to see the horrified look on their faces, because I'm like, if you want to know how badly these cells can be abused and still live, you will have that answer after we work on it for a while.
And Maggie, I'm thinking these cell line, the integrated sequences, those might be a great target for engineering. CRISPR?
Yeah, we thought of some research projects as well, just having, you know, students PCR amplify and take a look at the locus, you know, the insertion. There's going to be a paper coming out from Millipore Sigma that will give us more information on that.
So I know the Nimble Cho, they're doing a lot of that. Yeah, so.
Right, and so there should be an easy to set up PCR test. What we do know about NISTCHO, and I believe it's all anecdotal at this moment, there's a single integration. There's 3 copies, light and heavy and light chain, and it's very stable, which we didn't examine other than what Maggie talked about, but we'll know more soon. And it's been sequenced, and the sequence will become, the complete sequence will become available.
I have a question. Is there a null host available, a non-transfected null host available?
So that would be chosen, CHOZN.
Yeah, right. But you can get
$33,000 a vial. Yep, that's it.
sorry.
Part of the agreement when you get NISTCHO is you may not perform any engineering of the cells.
Right. I think it's just that's for some people wanting to look at, like, you know, host cell protein and things like that. There obviously there are reasons to have a null host, but maybe not at $33,000 a vial. Okay.
We're not looking for funding for that right now.
No.
I'd like to take this time to thank our presenters, Maggie and Laurie. We are just after the top of the hour. I will still stay online for those of you who didn't get all the links. We have a lot of links in our chat. Please complete our survey that Sandy put in the chat. There's the link to that. The survey helps us in planning future events. There are also links to the hackathon application, the Innovate Bio website, and also the link to future ATE project talks. Again, thank you very much to our speakers. That was a wonderful presentation, very informative, and we had a great turnout today. Best wishes to everyone for a great Friday and a safe weekend. Thank you.
Thank you, Dion.